Good afternoon. On behalf of the Lupus Foundation of Minnesota, I want to thank everyone for joining us today for this webinar called Using Anergy to Reestablish Tolerance to Self-Antigens. My name is Sandy Parnell, and I'm the Director of Education and Supportive Services at LFM. I'm very pleased to introduce our presenter, Dr. Daniel Mueller. Dr. Mueller is a professor of medicine and director of rheumatic and autoimmune diseases at the University of Minnesota Medical School. He is also a director of the autoimmunity program within the medical school's Center for Immunology. The major focus of his academic program is the investigation of the biological and biochemical mechanisms that underlie the maintenance of T and B cell tolerance within the peripheral immune system. Before Dr. Mueller begins his webinar presentation, I want to talk a bit about the functionality of the webinar system itself. At this point, your webcam function should be turned off, as well as the mute function should be turned on. The only voices you will hear are my own and Dr. Mueller's. Your audio will be heard through your tablet or computer. We invite questions during the presentation. All you have to do is type in the question in the sidebar. I'll see it, nobody else. And I'll ask Dr. Mueller the questions during or at the end of the presentation. Also, we're recording today's session for future audiences. The recording will be archived on our website. At the end of the webinar, you'll receive a survey in your email. We hope you take a couple minutes to complete the survey because your feedback really helps shape our future webinar programming. And finally, if you would like a certificate of attendance, just email me at sparnell@lupusmn.org and I will send you that certificate. So without further delay, welcome Dr. Mueller. We look forward to your presentation.
Thank you, Sue. I hope everybody can hear me okay. Today, I'd like to talk to you about some experiments that we've been doing that I think are relevant to the question of what's the best way to treat autoimmune diseases such as lupus. And in fact, the work that we do in our laboratory is to understand the mechanisms that can take autoreactive B cells and T cells and really turn them off. And that would be one approach to selectively eliminating those responses that cause disease in patients with autoimmune disease, while allowing the B and T cells that are protective to us and that fight infection, while allowing them to continue to operate normally. And so that's really our long-term goal in that. And today what I'm going to do is I'm actually going to show you the results of experiments. This is a mixed audience, some of which are going to be scientifically savvy and some of which are going to be lay people. And while my slides are going to be very scientific and you're going to have a chance to see the data just like any scientist would, I would like to kind of talk in terms that anybody could understand. And so if there's any questions that you might have to clarify something that I've said, I want you to go ahead and type the question in and I can respond to it during the talk. If, on the other hand, if you'd like to understand more and sort of delve a little bit deeper into some things that I've said, I'd like to save those questions to the end of the session and we can address them at that time. So one thing I'd like you to do is if you have a pen and a piece of paper, go ahead and get that out, because at some points during the talk I'm going to just have you write a few things down so that you can follow along when I'm using some of the acronyms that are very common to immunology, since this is an immunologic talk. So let's go ahead and get started with the first slide. The first slide really is just a model slide that I published back in 2010 that described the results of decades worth of work on the immunology of T cell tolerance. And while I'm not going to go through the individual molecules here that sort of determine whether a T cell is going to be aggressive and cause autoimmune disease, or whether a T cell could be rendered tolerant to antigen and really be put into a state where it can't hurt anybody, I do want to point out the fact that one of the most important things that determine whether a T cell is going to be what I call immunogenic, and that's a T cell that's responding to antigen, the target. It's responding to that in a very forceful way. The determinant of that is when there's an infection, and that's important because we want to be able to fight infection, and infections are very good at stimulating the antigen-presenting cells, which are the kind of the hairy-looking cells on this slide, to activate their nuclear factor kappa B or NF kappa B. And once the antigen-presenting cells are in that NF kappa B active state, then they can drive T cells to be the CD4 T effector memory cells that protect us from infection. In contrast to that, if antigen-presenting cells, and again, these are the cells that pick up targets in the immune system, and those targets that sometimes, in fact, are our own tissues. These cells pick up bits of our tissue. Luckily, because those bits of tissue aren't associated with any infection, those antigen-presenting cells don't upregulate NF kappa B, and they instead drive the T cells to become what I call anergic. An anergic really represents a T cell that can't respond any longer. It really is left in a state where it's just going to sit in the immune system and leave things alone. So, in fact, in patients with autoimmune disease, we'd like to be able to identify those T cells that are causing disease and put them into this anergic state so they would leave us alone. And that's really the goal of our research. Let's go to the next slide. So the question that I want to start with is, can we find any evidence that anergy, in fact, is a natural part of our immune system function? The previous model, the cartoon that I just showed you, is really based on a lot of experimental work that was performed either in tissue culture dishes or using T cells of very defined specificity that have been engineered to respond to autoantigens. And we're actually trying to move this work into a more physiologic setting where the T cells are more like the ones that respond to infection or that we think respond to autoantigens. And so the first approach that we wanted to take to model this was to identify T cells that respond to fetal antigens during pregnancy and see whether or not those T cells are put into this anergic or unresponsive state. And as you all know, the fetus is, in some ways, it's different than the mother. It contains genes and antigens that are derived from the father. And so the mother's immune system needs to learn to tolerate those new antigens that are carried with the fetus. And so we think that, that this could be a model for teaching autoreactive T cells to tolerate the self-antigens that are targeted in diseases like lupus. Now, this particular experiment is done in mice. In fact, all these experiments I'm going to show you today are done in mice as sort of a precursor to carrying out these experiments in humans. And so the way that we designed this experiment was to take a garden variety mouse, but insert into that mouse using molecular genetic techniques. Insert into that mouse a new antigen. And if you have a pen and paper, the antigen that I want you to write down is called 2W1S. And this 2W or 2W1S antigen, in some ways, it's modeling antigens like SSA or double-stranded DNA that might be the targets of disease in patients with lupus. And so it's just in this particular animal, it expresses this artificial self-antigen called 2W or 2W1S. And so we're going to monitor T cells that are specific for this particular antigen. And so we've taken males that have this transgenic antigen, we bred them to normal female mice. And so these mothers, these pregnant hosts then carry a fetus that in fact expresses this model antigen called 2W1S. And then what we do is towards the end of pregnancy, and in a mouse that pregnancy goes pretty fast, it's only 18 days. So at about 18 days then we take a look at the immune system of these mice and we see whether or not the 2W1S specific T cells have responded, and we look to see whether there's any evidence of tolerance. So we'll go to the next slide. And so this represents the type of analyses that we like to do in our laboratory. This is a flow cytometry analysis. And really all you need to know about this analysis is that we use some expensive equipment to sort individual cells of the immune system apart and be able to identify rare cells. And in this particular, in these panels here, what I'm showing you are little dots, and each of those dots represents the characteristics of one individual cell. And in this particular case, we're looking for T cells that are specific for this 2W1S antigen. And we can actually identify them by flow cytometry because we can offer all the T cells in the body of these mice the 2W1S antigen. And the antigen that we offer has a fluorescent probe, a tag on it. And then when we actually push these T cells through the machine can identify the presence of that fluorescent tag, and the T cells demonstrate then a different characteristic. And in this particular experiment, if you push the button one more time to the next slide, you'll see that we can identify in these pregnant hosts at day 18, went a little bit too far there. Go back one. At day 18, we can find these 2W1S specific T cells by flow cytometry because they were able to bind up the antigen. And what's remarkable about day 18 in these pregnant hosts is that there's a whole lot more of the 2W1S T cells around. So between the time prior to pregnancy and by the end of pregnancy, these fetal reactive T cells have been called out of the immune system and have been driven to undergo a response that increased their numbers and increased it more than tenfold. Now I'll go to the next slide. This is just a plot that demonstrates all the different time points and different groups that we've explored in this pregnancy system. And if you focus on day 18, what you can see is that there's a particular phenotype of cell called an anergic cell that increases in numbers specifically. And that's of great interest to us because those are the T cells that we thought would arise in pregnancy. And so this gives us very strong evidence that T cells can respond to fetal antigens by developing a pregnant state. And I'm going to show you in the next couple of slides what that state looks like. Go ahead to the next slide. First of all, if you look down on these T cells more, kind of drill down on them, at day 18 of pregnancy in mothers that were bred to a transgenic antigen-bearing mouse, what we can see is there's suddenly more T cells in that right-hand panel, the very right-hand panel where it's labeled 35. There's a large increase in cells that express a protein called FOXP3. And this FOXP3 protein is a master regulator for T cells that actually are important in protecting against autoimmune disease. So this is one piece of evidence that during pregnancy, any T cell that can see a fetal-derived antigen, there is the opportunity for that T cell to actually take on a regulatory phenotype that protects against rejection of the fetus. So that's a very remarkable finding. It's been seen before. We're not the first ones to see that. But we're the first ones to show in that panel that's marked 44, we're able to show that there's a large increase in cells that don't express FOXP3, but in fact demonstrate evidence that they saw that fetal antigen. And those are the ones that we postulated were anergic. Now go ahead to the next slide. So those are the Tregs. Now what I'm showing you here in the next slide is gating down, drilling down just on those T cells that demonstrated an increase in number and demonstrated a marker that suggested that they'd seen that fetal antigen. And now using a couple of other markers for the development of this anergic state. One is called folate receptor 4 and the other is called CD73. What we find is that just in these T cells that are responding to fetal antigen, the 2W1S specific T cells, that they're much more likely to develop this anergic phenotype than they are to develop that dangerous T effector memory phenotype. And this is really the kind of phenotype we'd love to have develop in T cells that are specific for self-antigens. We want them to be more anergic and less like effector cells that cause disease. So we were able to demonstrate this in pregnant hosts, demonstrated in T cells that are responding to the fetus. Next slide. And then again, hit the button just one more time, and it'll show a bar here that I thought, go ahead and go back one slide. Yeah, this just enumerates at day 18, once again, that there's a large increase in the number of those anergic cells during pregnancy. And this now satisfies that hypothesis that we had, that anergy can be a normal part of tolerance induction to fetal antigens. Next slide. So we've been interested in finding proof that these cells are in fact less responsive to antigen. They clearly accumulated early on in response to the antigen, so it's not as if they were immune to having their antigen receptors stimulated. And so in fact, when we began to look more carefully at these anergic cells, and they're shown here in the red circles, again, if you focus at day 18, the fetal antigen-specific T cells that have the anergic phenotype at day 18, they've lost expression of a molecule called Ki67. And Ki67 is highly associated with responsiveness of the T cells and proliferation. So this shows us that by day 18, these fetal reactive cells have lost their ability to respond with an aggressive proliferative response. They've shut down their cell division, and that's exactly what we want them to do. Next slide. Go ahead and hit it again. This slide actually looks at their ability to make a growth factor called interleukin-2. And once again, anergic cells were very poor producers of interleukin-2, and that compares to the fetal antigen-specific cells that fell into our effector cell gate that we think only recently saw that fetal antigen and hadn't yet developed that anergic phenotype. So this is a formal proof that we can generate T cells that respond to fetal antigens, but that lose the ability to make growth factors like IL-2 and lose the ability to undergo cell division. Next slide. So I'm just going to summarize the first part of this webinar, and that's that I think that this fetal antigen experiment has given us some interesting information related to the normal T cell response to fetal antigens. And what we found is that only a small number of conventional CD4 T cells respond to this particular fetal antigen. Some of those T cells become T regulatory phenotypes, so they have this FOXP3 phenotype. And those cells aren't dangerous. They're in fact, I think, protective to the fetus. However, there's a whole other population of cells that sees the fetal antigen and eventually makes up as many as 50% of all the fetal antigen-specific T cells in the body of these mice. And this particular phenotype appears to be anergic. These cells lose the ability to proliferate, and they lose the ability to make IL-2. So this is, I think, a demonstration that we can induce this unresponsiveness in mature normal T cells. And so what we want to do now is we want to see whether or not there's any evidence that autoreactive cells could develop this phenotype as well. Next slide. So the question is, do anergic T cells exist in the natural repertoire? Because we have self-antigens in our bodies, and the natural repertoire of T cells come in contact with these self-antigens all the time. So if anergy is going to work, we ought to be able to find some evidence of anergic cells naturally. Next slide. So we began to look at the normal T cell, and we call this polyclonal because these T cells see all sorts of different antigens. Some of the antigens are probably towards pathogens. Some of the antigens might be the toxins that we need to avoid, but some of the antigens are going to be targets of autoimmune disease. And so it's a whole wide array of different targets for these T cells. But that's what we wanted to do. We wanted to study T cells that are specific for lots of different targets. But we used those same markers we used before. Remember, we used FOXP3, folate receptor 4, and CD73. And if you kind of march along the top of this complicated graph, what you'll see is that we see some T cells, not very many, just 9% of the what we call the antigen experienced cells in that middle top panel have that. They're very bright for folate receptor 4. They're bright for CD73. They don't express FOXP3. They did express CD44. Those are all the characteristics that we predict would make them anergic. And in fact, if we drill down on that 9% of the cells that have that characteristic phenotype, we find that they don't express the growth factor receptor for IL-2 called CD25. And they don't express very well that Ki67 proliferative marker. So they do seem to be fairly inert cells. So that was promising to see that sort of a phenotype. Next slide. That's right. Next slide. And so what we did is we drilled down further on those cells to see whether or not they in fact could make the types of cytokines that aggressive, dangerous T cells make, including interleukin-2, like I mentioned before, and this other dangerous cytokine is called gamma interferon. And what I'm showing you in those lower panels is I'm showing you a comparison of T cells that have a T effector memory phenotype. So these are the type of T cells that might protect us from a diphtheria toxin, or they might protect us from tetanus if we're exposed to a rusty nail. If you look at those T cells, they in fact are very good at making IL-2. Almost half the cells make IL-2, and there's also a substantial proportion of those cells that make both IL-2 and gamma interferon. So those are good protective T cells that I think we want to have in our immune system. But if you look at those cells that we previously found to have an anergic phenotype, they weren't very good at making any cytokines. They didn't make IL-2 and they didn't make gamma interferon. And so that supports the notion that they've downregulated their capacity to make these cytokines. Next slide. We looked at a number of different molecules on these cells, and this is a different form of flow cytometry plot where we really just look at all the cells in what's called a histogram. And I think the most important thing that I want you to see here is if you look at that, the middle set of panels from the top, so the ones that are labeled anergic, in those light lines, you can see that for the molecule PD-1, the molecule CTLA-4, the molecule neuropilin-1, the line seems to be shifted to the right, suggesting that on average, they're brighter in expression for these molecules as compared to the dangerous T effector memory cell. And again, if this were an autoreactive host, animal, and perhaps even an autoreactive person, we would expect that the dangerous T cells that are causing autoimmune disease are in the upper panel, the T effector memory panel, where they don't do a very good job of expressing these molecules. All of these molecules at the bottom, the PD-1, the CTLA-4, the neuropilin-1, those are all molecules that tend to dampen the immune system. And so we like the fact that anergic cells overexpress these molecules and lead us to believe that if we could drive the expression of these molecules in the dangerous population of T cells, that we could gain better control of autoimmune disease. Next slide. Now, the expression of PD-1 and CTLA-4 and neuropilin-1 on these anergic cells was very similar to what we saw on those bona fide FOXP3 positive T regulatory cells. And again, FOXP3 is a good molecule to have on a T cell that's autoreactive. It keeps that cell from causing damage in the immune system and actually works to suppress autoimmune disease. So we like the fact that anergic cells express these molecules, but even beyond that, it makes us wonder whether anergic cells have a molecular relationship with FOXP3 positive Tregs. And so we looked for evidence of this by doing what's called a methylation study. And so we looked at the genes that in FOXP3 Tregs, we looked at genes that are frequently demethylated. And demethylation is associated with better expression of these genes. And so all the genes we looked at, TNF receptor superfamily 18, CTLA-4, IKZF4, these are all genes. You don't need to know what they're for, but it's helpful to know that these are all genes that dampen aggressive immune responses. They tend to quiet them down. And so Tregs tend to overexpress these genes and tend to demethylate the genes. And what we found is in the group of anergic cells that they tended to look more demethylated. They looked a little bit more like FOXP3 positive Tregs than either naive T cells or those dangerous effector memory cells. And so that led us to believe that perhaps there was not just a molecular relationship between anergic cells and Tregs, but maybe there is some more direct relationship where perhaps an anergic cell sometimes becomes a Treg. Next slide. And this is just one more gene that shows this sort of intermediate pattern for the anergic cells. Next slide. So to summarize this investigation for evidence of anergy being present in the natural repertoire of T cells, what we found is that amongst all these various T cells in the body of mice, there's a fraction of polyclonal anergic phenotype CD4 T cells in healthy hosts. And that's what's important there is that we think that the development of anergy in these T cells relies on a healthy immune system. If the immune system is predisposed to autoimmune disease or if it's damaged in some way, then we think that disease might develop, autoimmune disease might develop because of a failure to induce this anergy. These T cells that had an anergic phenotype were defective for growth factor production, and they seem to be defective for proliferation as well. Anergic polyclonal cells also showed high levels of these counter-regulatory molecules, PD-1, CYP4, and neuropilin, really very similar to FOXP3 positive T regulatory cells. And finally, when we looked at the genes that are frequently demethylated in FOXP3 positive Treg cells, these anergic cells, who themselves are FOXP3 negative, these anergic cells also demonstrated more demethylation, perhaps suggesting that they could be induced to be more Treg-like under the appropriate conditions. Next slide. So we wanted to push that question. We wanted to test whether or not anergic cells could ever give rise to FOXP3 positive Tregs, because that would be a wonderful outcome in somebody with autoimmune disease, to take the T cells that are actually aggressive and responding to self-antigens and provoking inflammation. If we could turn those T cells first into an anergic phenotype and then drive them to become a T regulatory cell, we think that those T cells would actually turn around and suppress the disease and allow those individuals to regain their health. Next slide. So we investigated this question of whether anergic cells could become Tregs by doing an adoptive transfer of these T cells into mice that lack their own T immune system. And a number of investigators had previously shown that transfer of T cells into mice that are genetically incapable of making any of their own T cells results in an expansion of that population of T cells, and that can include the FOXP3 positive Tregs. And so we wanted to see, well, what would happen if we sorted out a purified population of anergic cells and transferred them into what we call lymphopenic mice. So they're lymphopenic because these mice don't have their own lymphocytes, their own T lymphocytes. What would happen if we transferred anergic cells into these mice? Would any of these T cells become Tregs? Next slide. Go ahead, next slide. And so we did that. We transferred purified, I'm sorry, you can go back. We transferred purified anergic cells into mice that lack their own T cell repertoire. And then we waited 21 days, and then we recovered the immune system from these recipient mice and checked to see what had happened to the anergic cells. And if you look in the top panel, the anergic cells are the second from the right. We also included pure populations of Tregs. That's the rightmost panel there, as well as naive T cells, as well as T effector memory phenotype T cells. And we transferred each of these populations to see what would happen to their phenotypes. And the uppermost panels are really just looking to see whether they appear anergic. Now, the anergic cells going into these animals, about 2/3 of the T cells lost their anergic phenotype. So we know that animals that are defective in their immune system, that they are lymphopenic and lack their own FOXP3 Tregs, they have a very difficult time maintaining T cells in an anergic state. And we actually think that that's a risk for autoimmune disease development. There were about a third of the cells that seemed to maintain their expression of folate receptor 4 and CD73. If you looked at Treg cells, they had a somewhat similar phenotype, though again, they had about a third of the cells maintain expression of folate receptor 4 and CD73. Now in the lower panels, we stained for FOXP3. And again, FOXP3 is that master regulator of T regulation. And so any T cell that stably upregulates this FOXP3 molecule, that T cell generally demonstrates a suppressive or regulatory phenotype that doesn't cause autoimmune disease, but actually protects it against autoimmune disease. And the remarkable result from this experiment is that in a population of anergic cells that was transferred into these hosts 21 days earlier, at the end of that 21-day period, about a quarter of those cells now took on a FOXP3 phenotype. So that in fact is a novel finding that is very exciting and perhaps gives us a whole new opportunity to produce functionally that could suppress autoimmune disease in patients who have poor health because of these aggressive T cells. Next slide. And this just shows how good the anergic cells. The anergic cells were much better than either naive cells or T-effective memory cells at giving rise to these FOXP3 positive Tregs, as we call them. In fact, they were almost as good as Tregs themselves at generating this population. And so that is very encouraging because it perhaps gives us the opportunity to generate even more Treg cells. Next slide. Now we wondered whether or not these FOXP3 cells that we generate from autoreactive, that we generate from this anergic population, we wondered whether or not they work, whether or not they suppress disease. And from our study of these anergic populations, everything about these cells suggests to us that these anergic cells became anergic because they responded to some antigen. And we think they responded to self-antigens, meaning tissues in our own body. And that would fit with the idea, just like the T cells that responded to fetal antigen had become anergic, we think that these naturally anergic cells, somewhere in their lifespan, came in contact with a self-antigen and developed this anergic phenotype. And so if that's true, we think that they are potentially dangerous cells if they lose their anergy. And so we wondered if we prevented the cells from becoming FOXP3 positive Tregs, whether or not these anergic cells would threaten these mice after they lost their anergic phenotype. And so we were able to test this by taking advantage of some molecular genetic technology where T cells are engineered to express a receptor for a toxin called diphtheria toxin, but only express that receptor if they become a Treg. And so by taking advantage of anergic cells that are designed to express this diphtheria toxin receptor right at the time where they became Tregs, and then also taking advantage of diphtheria toxin, we're able to deplete any sort of anergy-derived Tregs that might develop over the course of 21 days. And what we found is that if we transfer anergic cells into lymphopenic hosts and allow them to undergo this anergy reversal, but at the same time we destroy any T cell that would become a FOXP3 positive Treg using diphtheria toxin, what we showed is that these T cells began to lose weight. And that's shown here in the open circles. And over 21 days, the average animal in this experiment lost 20% of its weight. And weight loss in experiments like this is frequently the result of the development of an inflammatory bowel disease and a colitis, because patients with inflammatory bowel disease and colitis tend to have, tend to lose fluids, and because they can't maintain their fluids, they get dehydrated and lose weight. And so that's what we think is going on in the system. So this this experiment gave us the impression that the FOXP3 cells that we generate during anergy reversal, that they're functional, that they actually suppress this weight loss, and we think that they're suppressing autoimmune disease. Next slide. Now, we have more evidence to suggest that, in fact, this isn't just dehydration, that this is, in fact, bona fide autoimmune disease. And the evidence that we have is that animals that developed the weight loss, so these were the animals that received anergic cells and then were treated with diphtheria toxin to deplete any Treg that would develop, these animals developed autoantibodies. And this is just a graphical representation. The dark red shows sera that was collected from one of these mice that was losing weight. We found that the sera contained antibodies that would bind either to the gut or the lungs, salivary gland, liver in various mice. These are antibodies that would be present and could bind to these tissues. So this presence of autoantibody is a very strong evidence that the destruction of Tregs in the system is leading to the development of autoimmune disease. Next slide. So to summarize this third part of the talk, the anergic compartment appears to be enriched in progenitors for FOXP3 Tregs. And this is a novel and I think remarkable finding, and opens up new avenues for the study of FOXP3 positive Tregs. We showed here some at least preliminary data that anergy-derived Tregs can suppress conventional CD4 T cell lymphopenia-induced proliferation and wasting disease in these mice. And then finally, anergy-derived Tregs suppressed the development of autoantibodies. And remember that in individuals with autoimmune diseases such as lupus, autoantibody production is a hallmark of the disease. And in fact, we think in some parts of the human disease, such as the kidney disease in patients with lupus, we think that the autoantibodies that are produced are in fact pathogenic, that they actually cause the inflammation in the kidneys. Next slide. So in this last part of this webinar, I want to address the question of autoimmune disease as it relates to arthritis more directly. And so illnesses such as systemic lupus, rheumatoid arthritis, scleroderma, other diseases, Sjogren's, they can have an autoimmune arthritis as part of, as one of the manifestations of the disease. And so we wanted to ask the question whether anergy can play any role in protecting against the development of arthritis. And so we're going to take advantage of an arthritis model system that we've developed in our laboratory and really ask the question whether anergic cells have any power over this development of arthritis. Next slide. So I want to first introduce you to our animal model of arthritis. And so you're looking there at the paws of a mouse. And the model system is really quite simple. We transfer into host autoreactive T cells. So these are T cells that are specific for a self-antigen called glucose-6-phosphate isomerase. Now that's a big long term for a protein, but I want you to imagine that that protein is just like any other autoantigen target that might be present in a patient with lupus or Sjogren's disease. And so these T cells are specific for that autoantigen. And again, if you transfer these T cells into mice that are defective, that have a defective component to their immune system so that they can't make their own Tregs, they are defective for T cell development. If you transfer KRN cells into those animals, at about day 10, we see that there's swelling, redness, and warmth of the joints. And I'm showing you in the lower panel, the lower mouse panel A there, that paw is sort of swollen and engorged and is beginning to sort of develop a deviation of the fingers that's consistent with arthritis. And you can compare that to the upper panel. Now the upper panel also got these autoreactive T cells, but that animal never developed any arthritis. And the reason is because that animal is a normal host and it's not genetically predisposed to arthritis development. And so the immune system is able to take those autoreactive T cells and cope with them. And in fact, I'll show you in a minute that those T cells become anergic. The panels B and D are just showing the onset of arthritis takes about 8 days for it to begin to develop, and then by day 10 it can become very severe in animals that, again, are lymphopenic, so they're defective because they don't have their own Tregs. And those same animals also lose weight, just like the other experiments that I've shown you. Next slide. Now, what's interesting about this particular model is that we can find evidence of autoantibody production in these hosts that develop arthritis. Now, remember that the animals first start developing arthritis at about day 8. We can see already at day 7 that there are B cells that have undergone a clonal expansion. So again, they've increased their numbers greatly in these mice, and that's shown on the lower right panel there. That there's a large expansion in that circle there of B cells that can bind to glucose 6-phosphate isomerase, GPI. And we're using a fluorescent labeled GPI to find these T cells in these B cells in the immune system by flow cytometry. So this again is, now we're looking at B cells. We're looking at the cells that produce antibodies, and in this case these are GPI specific B cells. Next slide. Now here, unfortunately, this got cut off a little bit, it looks like. So let's go ahead and look at the next slide. So the fact that these B cells are present, that's actually associated with production of autoantibody, though there was a mistake in that slide and I couldn't show you the production of autoantibody there. So in order to test whether anergic cells play any role in protecting against arthritis, we again did this transfer system where we isolated out anergic cells using flow cytometry, and we carried these anergic cells and transferred them into mice that are lymphopenic. And these are the disease-prone mice, the ones that get arthritis under our conditions. And we allowed our anergic cells to really repopulate these hosts, to really try to normalize these hosts to see whether the anergic cells could make these hosts resistant to the development of arthritis. And the anergic cells, in this case, are not specific for GPI. Of course, there may be some cells in there that are specific for GPI, but we don't know that there's any large number. We think that they're just specific for any number of self-antigens. And going into the lymphopenic host, the anergic cells don't express FOXP3, and therefore they don't immediately have any ability to suppress arthritis. But the question that we posed here is whether or not 21 days later in these lymphopenic mice, would the anergic cells undergo the conversion to FOXP3 Tregs, and would that then protect the mice from arthritis once we transferred in those dangerous KRN CD4 T cells? Next slide. So what this shows is the results of a large experiment where we analyzed the KRN T cells, the dangerous T cells, at the end of the experiment. So this was approximately 12 days after transferring the KRN cells. And so in the leftmost panel, this is really just a bar graph that shows how many of these dangerous CD4 T cells called KRN. These are the ones that respond to the autoantigen, and we think they interact with the GPI-specific B cells to cause disease. We looked to see how many of those animals, how many of those T cells would accumulate in the animals. And what you see is that a large number accumulated in the lymphopenic TCR alpha chain knockout. KO is knockout. Those are the animals that are getting sick. If we compare that to the anergic reconstituted group, they don't seem to accumulate as many of the knockout cells. Excuse me, they don't accumulate as many as the KRN cells. The dangerous cells are kept in check. And so that's consistent with the idea that Tregs developed in these mice. And in fact, if you look at arthritis in these mice, once we put anergic cells into the hosts, fewer of those mice developed severe arthritis. A couple developed A couple develop severe arthritis, but the majority either develop no arthritis at all or really quieted down the disease. So that leads us to believe that the anergic cells have some capacity to suppress arthritis. Next slide. This ability to suppress the arthritis corresponded with the ability of these anergic cells to undergo a reversal of anergy and to develop a FOXP3 positive phenotype. And what was interesting about this is that these anergic cells or anergy-derived Tregs then were able to force the KRN cells into an anergic cell phenotype themselves. And we think that's why mice don't develop arthritis. And so in a sense, anergic cells going into this defective host were able to restore the immune system, restore the population of T regulatory cells that's necessary to prevent the development of arthritis. And that prevention of arthritis is through induction of anergy. So in some ways, we think of this as sort of an infectious sort of tolerance. Tolerant cells can drive other cells to become tolerant. Next slide. Go ahead, next slide. So in summary, in normal host, GPI, oh, okay, go back one. There was a slow attack there on the button.
Is that the right slide?
There we go. That's the right slide there, yes. So in normal hosts, the GPI-specific CD4 T cells are induced into an anergic state. And tolerance is preserved. So those dangerous KRN CD4 T cells going into normal hosts, they don't cause arthritis because FOXP3 positive Tregs make them anergic. And once they're anergic, they can't proliferate anymore. They can't undergo cell divisions and increase their number. They lose the ability to make cytokines like interleukin-2 and gamma interferon. And they no longer can cooperate with the GPI-specific B cells to cause disease. And then in Treg deficient hosts, on the other hand, you can't get anergy to be induced. Now, anergy-derived FOXP3 Tregs are perfectly capable of suppressing these dangerous CD4 responses and appear to be able to prevent the development of arthritis. And so this is, I think, a model system for how perhaps in the future we'll convert anergic cells into Tregs to protect against arthritis development. Next slide. So at this time, I really want to just conclude this webinar and open it up for any discussions. And I want to list some of the conclusions here that I draw really from all the literature as well as from all of our experiments, not just the ones we've shown today. And we think that the evidence suggests that the development and maintenance of T-cell anergy, and we think this occurs after chronic exposure to self-antigens, we think that protects against autoimmune disease because it turns those T-cells off. It keeps those cells from dividing any further and becoming a big part of the immune system, and it keeps them from helping B cells become autoreactive. We also think that T-cell lymphopenia, or perhaps more selective defects in existing FOXP3 positive Tregs, we think that's an important risk factor for the development of autoimmune diseases such as lupus and rheumatoid arthritis. And in fact, in patients with autoimmune disease, a number of studies have suggested that Tregs are defective. And so it may be that this defect in FOXP3 precedes the development of inflammation and allows for poor anergy induction and expansions of dangerous autoreactive T cells. Nonetheless, in normal hosts, the body appears to maintain a population of self-reactive anergic cells, and we think that they may be a reservoir of Treg progenitors. And perhaps the immune system calls on these progenitors during periods of lymphopenia or Treg dysfunction, calls on these cells to become Tregs and to restore normal homeostasis. And maybe that's what happens for most individuals and allows them to protect themselves from autoimmune disease, but that there's some disadvantage or some disequilibrium in patients predisposed to autoimmune disease that renders this ineffective. So in summary, anergic polyclonal CD4 T cells may prove therapeutically useful to restore antigen-specific tolerance, and we think that this has the potential to cure autoimmune disease, because if you could take all those autoreactive T and B cells and put them into an unresponsive state, you wouldn't have that drive for autoimmune disease, and yet you'd still have all the other T and B cells that protect us from infection unaltered and still able to function. So we could move away from generalized immunosuppression and move to more specific and curative autoimmune disease therapy. And I think that's it. I want to acknowledge some of the people who have done this work in the next slide. Should be one more slide. There it is. This work that I showed you today has really recently been accepted for publication in Nature Immunology and should be out before the end of the year. It's work that's really been spearheaded by Lucky Kalikar in the laboratory. A number of the experiments have also relied on Nai Zhang, Shirley Schmiel, Ryan Martinez, Phil Titcomb, and Justin Taylor to carry out. And none of this work could have been done without the support of our advocates in the community as well as the NIH. We've had financial support from both the Rheumatology Research Foundation as well as from the Lupus Foundation of Minnesota. And then finally, much of this work is supported by a grant from the National Institute of Allergy and Infectious Diseases. Thank you. I'm going to open this up to any questions now.
Dr. Mueller, there are 3 questions, 2 of which can be, I think, collapsed. How far are we from actually promoting anergic cells to act as suppressors of autoimmune disease? And the second part is then, what would be the next steps of your research to get us closer to that point?
I think the next step is to identify markers that reliably find anergic cells in humans. And our work in mice has clearly identified these anergic cells in the spleen and lymph nodes of mice, but we don't find very many of these in the blood. And so the question in humans is we need to do a study that first looks at tissue from spleen and lymph nodes in humans, which is not that easy to do, find the relevant markers for these anergic cells, and then see whether or not there are effective ways to identify and capture blood anergic cells in humans. I think that's the first step, and that's really the work that we want to move to very quickly, and some of those experiments are underway. Then the next step would be to determine, are there effective means of promoting this conversion to FOXP3 Tregs in vitro, in tissue culture, because then we would have the opportunity to take these converted cells and transfer them back into individuals to see whether or not they can promote anergy induction in the dangerous autoreactive T cells. And then I think the final thing that needs to be developed before we can put this into motion is we need to find better probes to identify who the autoreactive cells are in humans. And we're doing experiments right now to identify the autoreactive B cells, and we have experiments underway looking at B cells. The T cell question is more complex for various biochemical reasons, but we have every reason to believe that we'll soon have technology to identify the T cells that are responding to autoantigens in patients with lupus and rheumatoid arthritis. And once we have the ability to identify those T cells, then we'll be able to see whether the anergy-derived Tregs are promoting energy induction in those T cells. And I think that's going to be coupling this identification technology together with techniques to create energy-derived Tregs. That's going to be a very powerful combination. And I think that those experiments are going to be underway within the next 5 years.
Terrific. And the other question is, what are your top 2 suggestions from people living with autoimmune disease, lupus in particular, to manage the disease?
Well, I think that in patients with lupus, very important management for them to start with is pharmacologic. We have agents that are available in the clinic that are frequently used, such as a good choice is something as simple as hydroxychloroquine. And while that is, it's still a generalized immunosuppressant in the sense that it's not specific for the autoreactive T and B cells. It sort of hits all the lymphocytes and immune cells. It's generally considered fairly mild in terms of its ability to suppress responses to dangerous pathogens. So that makes it very safe for patients with lupus. And yet at the same time, it seems to lock disease into place. It seems to keep the disease, seems to keep the autoreactivity from developing more and more autoantigen targets. And we know that the patients with lupus that develop more targets, in other words, begin to respond to double-stranded DNA or begin to respond to Smith or other targets, those are the patients that tend to develop the more severe manifestations of lupus, such as an example, kidney disease. So I think the first step to effective management of lupus currently is to have a rheumatologist and to be exploring use of some of the pharmacologic agents. And hydroxychloroquine is a great place to start. And of course, there's other agents available for generalized immunosuppression. In terms of other things that patients can do, I do think that the immune system is very sensitive to stressors in the environment, such as infection, other toxin stresses, emotional stresses. All of those kind of things are tipping the balance away from anergy induction to aggressiveness, aggressive T effector memory generation. And so if you're repeatedly exposing yourself to infections, if you're repeatedly exposing yourself to toxins, those are the types of things that I think, at least in theory, are going to make flares of lupus more frequent and perhaps make the spreading phenomena to increase numbers of targets more likely to occur. Now, I can't tell any individual what those toxins in their environment or what those infections are that they're exposed to, because it can be different for every individual. But I think common sense can help guide individuals in terms of avoiding some of the stressors to the immune system. The same thing is true for our diets. I think that there are some people whose diets are more rich in things like saturated fatty acids and poor in things like indigestible fibers, and those are the kind of things that are very immune stimulatory. And I think making sure that a diet is more balanced and has more opportunity to provide the GI tract and the microbiology that lives there with indigestible fibers, as well as omega-3 fatty acids. Those are all great ways to do your best to promote a healthy GI tract and promote good immune system health. I think those are all the best things that I can share.
Appreciate that. This concludes today's webinar. Thank you so much, Dr. Mueller, for the presentation today. We really appreciate your expertise in the area and the information we're walking away with. You clearly find this work exciting and powerful, and we wish you and your team the best of luck as you move forward with the critical research. Thank you very much.
Thank you.